Okay, and then just quickly going through that translation process again. Again, these are illustrations taken out of your textbook. I'm not going to ask you to restate this process of translation. I just want you to appreciate the definition of translation. I would be just familiar with the general steps in the translation process, in the generation of a particular sequence pair, and how proteins are synthesized through this process. For bacterial genes, for the expression of for bacterial genes, which differs just a bit from the biological system or the human systems, the cells will conserve energy by making only those proteins needed at times. Now, the human system or the biological system basically does the same as well. It will conserve energy as well. If a gene produces a product at a fixed rate, and we see that sometimes again in the human system as well, that particular types of proteins are synthesized at a fixed rate because it's required to carry out a particular operation. And so because of that, we call it constitutive, that these organisms will produce, or the genes will basically produce a product at a fixed rate. You should be familiar with that definition. Now, the enzymes we see are expressed in a fixed rate. These enzymes are expressed, need to be repressible genes, for example, that control certain types of gene expression, and inducible enzymes as well. An inducer basically is a substance or a substrate whose presence results in information or an increase in the amount of an enzyme. Such enzymes are called inducible enzymes. Genetically controlled response is termed as enzyme induction. Lactase production is in response to lactose being available in the system. Lactose is a complex carbohydrate, sugar that's produced, and lactase is an enzyme, a protein production that's produced at pretty much a constant rate for substances such as complex sugars such as lactose. Genetic regulation that decreases enzymes synthesis is referred to as enzyme repression. A protein that mediates these types of repressors. Repression occurs if cells are exposed to an overabundance of a particular end product in a metabolic pathway. We know that basically as feedback or feedback mechanism. Again, proteins that mediate this as a repressor. It occurs typically when there is an overabundance in a particular feedback product or an end product. And then feedback inhibition is the term that we hear referred to as often. Protein synthesis in bacteria is controlled in a system called an operome model. You should be familiar with the operome model, and just by definition refers to protein synthesis in bacteria. Remember, now protein synthesis occurs in all biological organisms, especially in the human system, but the model that's used to identify protein synthesis in bacteria is the operome model. 3 enzymes involved in the uptake and utilization of, say, the sugar, the complex sugar lactose, and this is in a bacteria referred to as Escherichia coli, which you're familiar with already, been able to identify that on the exam. The gene for these enzymes, structural genes, are close together on the bacterial chromosome. There's also an operator site that's next to the structural gene, and it's more remotely located to the gene that codes for the repressor protein, as we talked about earlier. The operator and the promoter site plus a structural gene is referred to as the operome. And again, this model is specifically for protein synthesis in bacteria. So there's an operator site, there's a promoter site, and then there's a structural gene. This is what's referred to as the operome. That's a perfect multiple choice question. Operator, promoter, structural gene, are all of the above. Yeah, I remember that. Somebody mark that for me. Send me a. Now, the lac operome, again, is what this model refers to. Utilization, production of a particular protein, lactase, which is going to be able to help in the catabolism of lactose. When lactose is absent, a repressor protein prevents the operator from making a lactose utilizing enzyme. Now that makes sense when you think about it. And again, this is a model. We're only talking about lactose. We're only talking about E. coli. But this is a model, and you can use this same model when we look at bacteria and we talk about protein synthesis. So yes, I do want you to know lactoferrin, but appreciate what we're talking about. We're talking about a system of protein synthesis, a system that you can apply to almost every bacteria in the environment. So this model kind of shows us and demonstrates that, how all of the various stages of this process works. So when lactose is absent, there's a repressor protein. Repressor protein will keep the enzyme, the lactose utilizing enzyme, from being produced. Small molecules referred to as co-repressors also required to bind at the repressor site. Not as important for you to appreciate. When lactose is present, there's some diffusion or diffusions into the cells. We see it will bind the repressor protein. Remember what happens when you bind a protein. If you remember when we talked about enzymes, if we bind a protein at a particular site, it changes the shape of that particular protein or that particular enzyme. When it changes the shape, it can no longer bind to the substrate. So what you see here, when lactose is present, we see that some will diffuse into the cell. It will then bind the repressor protein. Remember, the repressor protein actually stopped the lactose utilizing enzyme. So because it binds to the protein, now we see it cannot bind to the operator site, and thus that utilizing protein will be produced. The operator then induces the structural gene, as I just said, and produces the enzyme that can utilize lactose. This is an example of an inducible type of an enzyme. Again, this model applied to almost every bacteria and almost every protein that's synthesized in these various bacteria. Many genes are not regulated in this manner, but they're constitutive, just as we've talked about before. Usually represents the functions needed for major life processes. And we see this process especially in the biological system when we talk about the utilization of glucose and enzymes that are involved in the breakdown of glucose. Now, in the glucose effects, cells prefer glucose to lactose, but if the level of glucose is too low, the cell will respond with the cellular alarm signal. These are called alarmones. I didn't make up that name. Alarmones, which are basically cyclic AMP monophosphate adenosine triphosphate you're used to as a energy compound, adenosine monophosphate cyclic, which just means that the structure forms a octagon type of a structure which is closed. But this is an example of an alarmone. It's produced when glucose no longer is available, and it's required in the beginning of the changes in the operone system for the use of lactose. Other sugars use similar mechanisms. Inhibition of metabolism and alternative carbon sources when glucose is present is basically called catabolic repression, or the glucose effect. Again, preferentially, organisms want to use glucose. If glucose is not available, it's not being produced, then it may require the use of other substances as well, such as lactose, but then the process by which the enzymes have to be coded is particularly different. I believe that's all we needed to do in this. This is just an illustration from that same model that just shows you the utilization of glucose versus lactose, and how the number of cells grow much more rapidly in the presence of glucose than we see with lactose, because the enzymatic processes of many organisms, microorganisms, is set up more readily to appreciate glucose. There's different stages, chemical steps that are required for the utilization of lactose, so you can see how the regulation and the expression of the gene and the production of the total number of cells certainly is much faster in the presence of glucose versus lactose. And you can see in this particular organism, you see a change. You first see the utilization of glucose and how steep the number of cells will grow in a logarithmic manner. We've talked about the log phase already, logarithmic growth. But then what happens when it switches over to the utilization of lactose? as an alternative source because all the glucose has been utilized in the microorganism. So there's a period of time where no growth will continue. Now notice there's a plateauing here referred to as the lag phase, and then growth will start again when the microorganism changes now its phenotypic expression. It's now going to start producing the utilizing enzymes or utilizing proteins specifically for lactose, and lactose will be generated, and then lactose will be utilized.